I'll start. Right? It's already it's been 4 o'clock. So it's my pleasure to host as part of the DDD conference, the Desert Drylands Desertification conference. We are in theory in Sde Boker in the Israeli desert. So we're having a session about water flow in the soil plant atmosphere continuum, and we brought 7 awesome speakers. So you can see kind of the timeline, and to the speakers, I urge you to keep this this schedule, okay? Because I can't really mute you, and I won't, but try to make it on time. So just to kind of orient you of who we're going to speak about what. I'm by the way, I'm hosting this session with Naftali Lazarovich, Professor Naftali Lazarovich from Ben Gurion, and we have today. Oh, okay. So we're going to start with Tony Rockwell, and Tony going to give us kind of an overview on the SPAC, the soil plant atmosphere continuum. Okay, so he's going to take us from the soil through the roots and up above. And I guess he will ask all the difficult questions, probably very basic ones that we still are failing to answer. And then we'll have Andreas Kenma and Nimrod Schwartz that will show us some visualization of roots down there. Andrea Carminati and Celia Rodriguez Dominguez will take us from the soil root interface and up the tree. Where
we hold on for 1 second.
Yeah.
Please, all of you, turn off the browser. Or close the browser, Internet Explorer or Google Chrome, whatever you are using, or mute the video that you have there. Just pause it, because otherwise we will have echoes and people will hear several voices with delays, and it will be hard to hear the presentations.
Okay. So after Andrea and Celia, that will start from, we said, probably the soil root interface and take us up to the stomata. Then we will have Craig Brotherson. He will speak about shoots and visualization methods, both CT and MRI, and how he can try to understand the heterogeneity of the pressure within the xylem. And last, we'll have Jonathan Sorek, actually a PhD student from my group, that will speak about xylem cavitation in leaves and the plasticity of it. Okay, all of the people, by the way, or most of them, kind of use a novel visualization technique that really, I think, took our field a step forward. So, Naftali, you want to add something before Tony?
Yeah. So basically, when we are meeting soil scientists and plant scientists, it's a challenge. We have different units, we have different vocabulary, but those sessions are really important since we can interact, we can bring new knowledge, we can see what other people are doing. And I think that visualization and those non-invasive methods are really, really important, because they can shed light on processes that it's very hard to measure in usual quantities. So let's see what people are doing.
Great. So, Tony, you want to take the stage and do your thing?
Okay.
So I'm going to stop. I'm going to stop sharing. Sorry. Oh.
Why is that? Okay. So, okay, can people hear me?
Yes.
It says sharing is paused. I don't know, is that the case? Or you can see my title slide?
Everything is great. Just continue.
Okay. Oh. Okay, this is not working as I thought it would. So let me just quickly switch.
And I still see Yuri Hochberg's screen.
Yeah. I seem to have lost my mouse.
Tony, we can see your screen and we can hear you.
Okay, then I'm just going to start. Some people see Yuri's screen apparently, but what can we do? Okay, so I'm talking about perspectives on the flows of water and energy in the SPAC. And the SPAC is the soil, plant, air continuum. And the question I'm asking is, where do we stand? So you can think about that question in two ways. One is sort of what's the sort of current state of the field? What do we know? What do we not know? What do we want to know? The other is sort of a more methodological question or a philosophical question even, is where do we locate ourselves in the system to observe the phenomenon that we want to observe? And I'm going to kind of try to illustrate that with an example. Back from kind of the 1970s and 80s, there was kind of a controversy between physiologists who, I don't have a pointer. This is really going to be hard. Okay.
Wait, so Tony, maybe you'll start. People are claiming that they have a problem. Maybe you want to stop screen sharing and then restart it or something like that.
Yeah,
try that.
I seem to not. Try that.
Okay, we can see the mouse now.
Okay.
Ah, okay.
Just keep it like this.
Go back
we
here.
can see it.
Okay, I see that's not working. So something about playing the slideshow. So we'll just do it. We'll do it like this. Okay.
Good.
So clash of perspectives. There's a physiological perspective where, transpiration is just a product of a proportionality, which is controlled by stomatal conductance and boundary layer conductance, and a driving force, which is in this case, I'm using the mole fraction gradient of the leaf, the sort of the saturated mole fraction at that leaf temperature, and the difference of that value from that of the ambient air, okay? So this gives you kind of a snapshot. It's a steady state statement. It doesn't tell you anything about how the system is going to evolve. It doesn't tell you what's going to happen to the mole fraction of the atmosphere as a consequence of water being transpired into it. And it obviously doesn't say anything about what's going to happen to the soil as you're removing water from it. But really here, the question was about what's happening in the atmosphere as plants transpire into it. So meteorologists tended to take the perspective of coupling energy and water transport together. The advantage of that is mathematically, without any kind of compromising of the physics, you get to eliminate this surface temperature. You still have this question of where do we measure the humidity, if you will, of the ambient air, and what's the appropriate form of your transport conductances. So there's differences between laminar boundary layers and turbulent boundary layers, that kind of thing. To try to get away from those complicating factors, it was natural for these atmospheric physicists to consider limits to these equations. So in the limit that you have kind of a semi-infinite atmosphere in which you're evaporating into, and there's no lateral wind flow or anything like that, you can eliminate the ambient humidity. You can eliminate these transfer conductances or resistances, and you end up with a really beautiful description of the sort of the proportion of radiant energy that gets turned into latent heat transport and so evaporation. So which of these is correct? Is radiation controlling transpiration or stomata controlling transpiration? And of course the answer is it depends on the details. And it's just two different ways of looking at the system. And so we can use simplified models like these to look at our system, but we have to be mindful that we are using them appropriately and that we are looking at things in the appropriate limit. So today, there's sort of a variation of this controversy going on where you've got a group led by Novick, and they're taking a very physiological view of the question of whether VPD or soil drying are the biggest problem going forward in drought. Which of these is going to stress plants more. So they take a physiological perspective and of the effect of VPD on stomata. They sort of look at some sites to develop this relationship. They look at the kind of the reference levels to stomatal conductance as a function of soil moisture. So they're kind of painting this physiological picture. And the conclusion they come to is that VPD is more important than soil moisture. Now, another group takes a very different perspective, and they're actually using coupled models of the land and atmosphere interactions that are fully coupled. So this is a completely, physically, a totally complete description of the system. And they do modeling experiments where you eliminate the coupling and you see what happens. And what they find is that, in fact, both in the satellite data and in their models, VPD, extreme high VPD events are highly correlated with low soil moisture. So essentially what's going on here is that, yes, high VPD events do stress the heck out of the plants, but you only get those when the soil is very dry. And part of the reason why you get these high VPD events is that plants aren't transpiring in the first place, you see? So it's a coupled system, it's a cycle, and you have to be mindful of those feedbacks. Okay, so now we're going to play this game of perspective and where do we put ourselves and how do we, what kind of simplified models can we rely on? We're going to play this game in the soil a little bit. And I know there are others who know a lot more about soil who are coming after me, so I'm kind of sticking my neck out a little here, but I hope it's at least a kind of a useful introduction to what's coming. Okay, so the simplest possible model, right, of the soil is just a bucket. And what does a bucket do? It has a volumetric water content, and it gives up that water as the tensions grow. This is actually the model that those big land surface atmosphere models use typically to derive some kind of function of surface conductance. And at the physiological scale, they became very popular when we were all super interested in variable stem hydraulics due to things like cavitation. So Sperry really popularized this. I know Carminati has talked about this, and so I won't belabor the point, but there's an idea that if plants maybe increase their rooting density to the point where they can access every soil grain and suck the water off it, and water never has to actually flow through the soil. So the soil as a resistor doesn't exist, it's just a bucket. Okay. So does this model ever work? And when does it fit? And to answer that question, or to ask that question, I'm going to look at something people are now calling water potential curves. So these are plots of midday versus pre-dawn water potential. And Nipfer et al. From the McElroy lab at UC Davis just published a really wonderful paper in Plant Phys producing a lot of these. I mean, there's a lot of measurements, it's a lot of work. And I think actually there's a lot we might be able to learn from these. So we look at the two ends of these plots, it's pretty simple to see what's going on. Down here, it's super dry. Midday pre-dawns are basically the same thing. There's no transpiration. The roots have to be sort of at soil water potential. The whole plant is basically at soil water potential. Nothing's going on. That's fine. At the hydrated range, it's a little more interesting. You see that the middays are offset from the one-to-one line by a fairly constant amount. So what does that mean? That essentially means that given sort of some average conditions or average VPD and sort of average stomatal conductance, plant sort of the conductance of the SPAC is essentially constant, and you get a very predictable water potential drop between pre-dawn and midday. And here we're assuming that pre-dawn water potential is telling us the root zone water potential at midday. So that assumption and this sort of constant conductance through the whole SPAC works really well up here. This is in almond trees up until about a megapascal of tension. And then everything gets weird. So what's happening here? Okay, so in this paper, Nipfer et al., they sort of suggest that the point where midday and pre-dawn converge, that's sort of turgor loss. They fit a curve, and they argue that this break point here has to do with stomatal closure. In a commentary accompanying this article, Charrier takes a somewhat different view, and he identifies this region as anisohydric, this region as isohydric, and he shows how he expects certain physiological processes to relate to these different phases. The thing here that jumps out at me is he has the petioles cavitating even as the stomata are shutting. I think that's a little controversial. I'm not sure everybody would agree with that. But in any case, this is the effort, right? We want to match physiology to the behavior, the different phases we see in these diagrams. Okay. So ISO and ISO. I'm not going to get too deep into this. Yuri has a wonderful paper and tips about how this is not just a behavior of plants, it involves the soil. And we're going to see how that plays out in a couple slides. But there is a problem here with, there is something interesting about strict isohydry that I would like to get on the table. And that is that it's non-physical. It's impossible physically. It requires chemistry. So something like hormones, right? Why do I say that? Okay, so here's a guard cell pressure versus stomatal aperture curve from Franks. He built this curve with a pressure probe. But the point is that when guard cells are very turgid, they're not very sensitive to changes in water potential, right? This aperture it's in a nice plateau. So guard cells can lose a megapascal or two of turgor and nothing, and stomatal aperture doesn't necessarily respond. This is anisohydric, right? So that gives you this nice early part of the phase. Now, things get drier, stomata all of a sudden become much more sensitive. So this could be captured by this kind of behavior where you've got, well, that's something else. You've got, yeah, I eliminated one of my little figures here, that's okay. But so that gives you kind of a slope here. But you can't get a completely flat relationship between midday leaf water potential and aperture from this kind of curve. You need a step function to do that. And that means that stomatal conductance is multi-valued at any particular leaf water potential, which of course is impossible with a hydraulic feedback, and you have to invoke hormones. Now, if you plot the same sort of flatness against the pre-dawn, you see that it's, you get rid of that step function. And so that's the motivation for looking at pre-dawn and saying, okay, what stomata are really responding to is pre-dawn water potential, or roots water potential, midday root water potential, and not leaf water potential, right? So this motivates all of that root signaling discussion in a way. Okay. So which is it? A fundamental question we now have to grapple with is, what is the relationship between pre-dawn leaf water potential and root zone water potential at midday? Is this assumption that they're kind of the same that we typically want to make when we look at these plots? Is that assumption a good assumption? All right, so pre-dawn water potential. Let's say the soil is truly, the whole system, the SPAC is in equilibrium. What do we expect? This is something I never really thought about. I was totally shocked when a hydrologist kind of pointed this out to me. So maybe you'll find this shocking, maybe it's obvious to you. But anyways, here we go. Essentially, it's equilibrium, so water potential is zero everywhere. Because at the water table, it's zero, right? And equilibrium means no variations in water potential. Therefore, water potential is zero everywhere. That means that all gradients in tension follow the gravitational gradient, which is 0.01 megapascal per meter. There's no variations here. I mean, it's like, over 30 meters. What are we talking about? Maybe 3 bars or something. It's not a lot. we certainly see pre-dawn water potentials that are much more negative than one would expect. So, okay, the soil is just never actually in equilibrium, right? Generally, what's going on, even at pre-dawn, when the plants are not transpiring, is that there's some kind of flow from deep groundwater. And is there always deep groundwater? To ask a hydrologist, I don't know. maybe it's just rock. Who knows? But I usually think there's some kind of water table. So there's some kind of flow from that water table. Then you have your root zone. We have sort of drying around the roots, and we have a gradient in water potentials. And maybe, you know, this sort of zone right near the root, maybe that is being reported by dawn, assuming things like nocturnal transpiration, et cetera, are off the table. Okay. But, you know, the plants aren't transpiring, and yet the soil's out of equilibrium and there's a flow going on. What's going to happen when the sun comes up and starts to pull on that plant? Right? The root zone is going to get more out of equilibrium. So, a lot more? This is the question. So there's a really wonderful paper by Ian Cowan, the Australian, who sadly is no longer with us, but he solves this problem in a finite domain. And what we have here is a 4-day sequence. This is the time-dependent model. This is the full model, where he's giving us here the average of the root zone, which is this sort of finite domain, which starts out with some water level and it's going to fall as the plants transpire. Then we see what's happening at the root surface sort of over the course of this day, and we have leaf water potential. We're starting out at pretty mild potentials, and we can see that during the day, the root zone is getting drier than, well, considerably drier than the average, and then it relaxes back. So this root zone potential, one way to think about it is it's kind of like a spring. It gets stretched during the day and it relaxes back at night. Whereas over the course of these 4 days, on average, the whole soil is behaving like a bucket. It's just a monotonic drop in water potential. But of course, what really matters to the plant is this root zone. And then Cowan has that sort of strict psi-min. It doesn't have a stomata function, so that's why you get these flat lines where this is where the stomata start to regulate. So you can see that happening here. And there's one other thing I'd like to sort of point out while we're on the slide, which is that even when things get really dry and the stomata are really regulating, there still are these brief periods in the morning where the root zone has recharged overnight and the stomata are able to fully open. Right, and that's what we see. In fact, right, when you have kind of droughted plants, the way the drought progresses is that the stomata kind of they try to open in the morning and then they shut, and that sort of period where they're open and the degree to which they can fully open gets kind of smaller and smaller as the drought progresses. Okay, now. How does rooting volume affect this? This is kind of an interesting little sort of aside. So we have 4 different it gives us 4 different densities of rooting volume, sort of from less to a lot. And what's interesting is that I don't think rooting volume shows up in those pre-dawn midday plots. Like you don't see a signature of rooting volume in those. You only see it when you're considering the time domain. Because what happens is that if you're on this large rooting volume curve here, what happens is that you maintain, you basically are able to keep your root zone wet for longer, you transpire more, so everything's better. There's nothing better, except that at some point you crash and burn. So everything falls quickly once you've extracted sort of the whole soil and you've pushed the whole soil to the point where it's giving up all its water. Whereas if you have a low rooting volume, you have a lot more time and you get a much softer landing. So that's something to think about. And it's also something to think about if you're doing if you're working with potted plants and doing drought experiments, because, you know, if your pots are root bound, it's impossible to do that experiment because the plant's just going to go and then it's just going to boom. It's going to be. So be very careful about your rooting volume. Okay. So what do we expect to see in these midday versus pre-dawn plots if Cowan's right and we're getting this kind of diurnal pulling away of root zone potential from the bulk soil? In other words, pre-dawn is not root zone water potential. So I basically took Uri's model from the TIPS paper. It's sort of a simple hydraulic model. I added in a function, and this is going to be for grape because I found this wonderful paper by Levin and Williams and Mark Matthews from UC Davis, and they have this beautiful function for stomatal conductance as a function of midday leaf water potential. It's a thing of beauty. I mean, you never see data this clean. And the reason it's so clean is it's 17 cultivars, 2 years, and almost 800 measurements. You know, there's a lot of things in physiology where I just don't think we measure enough. Yeah, it's like, okay, anyways. So we have this nice function. Alec Levin was kind enough to share with me some unpublished pre-dawn and midday data. So I put this into the model. We have a function of soil conductivity, really soil conductance in this case, because anyways. And that's gonna, this is a sort of the classic curve. I took this functional form from Gardner. We're using an exponent that's suitable for clay, and I use a low range of the values for the parameter A. I don't know what the physical meaning of A is, but it is important. So what happens? Okay, so this blue line is what's happening at the root surface, and the black line is midday leaf water potential. And so what we see is that as your conductance and your water content start to crash at around half a megapascal, the root water potential is being pushed off the pre-dawn line dramatically. That is an inexorable push. I mean, look at the steepness of this curve. There's no way to avoid that. The soil is going to push you as negative as you need to go until your stomata regulates. So here we have the stomata at the point of this arrow is your 50% closure point. That's pushing this curve back towards the one-to-one line. So this is what's going on. These are the forces that are acting on this curve. So just to say this parameter A, if you make it bigger, let's see if I got this right. If you make it bigger, you get more anisohydric. Well, basically the soil stays closer to this and you have more anisohydric behavior. If you make it smaller, then everything gets pushed to the right and it just, your plant basically just can't keep its stomates open. So that's that. But the important point here, I just want to draw your attention to are the ways that this curve is shaped by characteristics of the soil and to some extent by the stomatal conductance. So the really important point is that it doesn't matter where you put the stomatal conductance. I mean, it does matter, but the soil, this line here will drop to force you to close pretty much no matter how negative you put that. Okay. So I fit this, I scaled the conductivity function for the plant to fit the data at the wet part of the curve. Wow, I am out of time. And so the soil is the only really fitted thing. So the question is not like, is this numerically correct? It's just, can this kind of simple model explain the pre-dawn midday pattern? And I actually think it does a pretty good job of explaining Alec's data. Okay. So can you guys give me 5 minutes or am I?
Tony, you're past your last 5 minutes. Try and finish in 5, okay?
I'll try to finish in 5. Okay. All right. So there is a question though in this model, and there's a little bit, there is kind of a bucket here, which I'll try to explain to you. So in a steady state model, we take as the size of our domain, it's basically just half the distance to the next root. But that means that in steady state, we have these sources that are providing the water that's transpired located in between all the different roots. These are all the cross sections of the roots, and these are the water potential isosurfaces around them. And a steady state model kind of demands that you have these water sources all around. In a way, it's a little broken, is what I'm trying to say. And what you really need to do, unfortunately, what the full model of the soil looks like is you have your roots with some spacing, and in this case, they're square. I apologize for square roots, but you see that the shape of the gradients is correct. This is the gradients in water potential. And then you have a water surface down here. That's, see, we have two different contrasting geometries is the problem. We have this kind of linear geometry, and then we have the circular geometry. It basically means that you have to solve this numerically. Now, we have solved this problem, or at least I've solved this problem in the context of plants. It turns out it's the exact same problem as vein spacing and a transpiring surface. It's just really the inverse problem. And that brings us to leaves, where I was going to talk about then how we model leaves. I'm just going to skip it, except to say this model is a little broken because this is the classic K-leaf Ohm's law model, if you're familiar with it. The problem with it is the water potential is a bulk leaf water potential, and yet in the model, it's supposed to be stomatal water potential. How big of an error is that? So I solved the full thing with coupled heat and mass transport. I allow for vapor transport in conjunction with the liquid. And we end up with a difference of about 8 bars between this bulk leaf water potential and the stomatal water potential. So you could drive a bus through that gap. So I would suggest that this is part of the reason why our stomatal conductance and midday leaf water potential curves are so messy. Is in fact, the models just aren't very good, and there's a lot of potential variation that sneaks in because of that. So okay, so I can add in stomata and reconstruct the gradient. So we can get this really beautiful complete picture of the leaf now. We're in good shape as far as leaves go. There is one thing I will alert you to, and that is work from Farquhar and Cherny-Sack, and they are challenging the idea that leaf surface, that basically that the water inside the air spaces of the leaf is saturated at leaf surface temperature. I think some of their data for these gymnosperms, conifers, indicates relative humidities inside the leaf of as low as 80%. That's minus 30 megapascal. This is impossible unless you're getting some kind of crust forming inside the leaf or something. So something's going on here. We don't know, but stay tuned on that front as far as leaves go. Okay, and with that, I'm finished. Let's say thanks to current members of the Holbrook Lab. This is what we look like these days on Zoom. Some funding, NSF, Air Force, and Harvard keeps the lights on. Thank you all very much. Start sharing.
Thank you. We're a bit behind schedule, so let's do one quick question. Just unmute yourself and ask.
Okay. There's nothing in the chat, so just move on, Yuri.
Okay, I got a lot of questions actually, but perhaps too many, so we'll do that later. So let's keep moving, and we have Andrea. You wanna share your screen, right? Andreas Kenma is.
Yes. Good afternoon, everyone. Oh, you actually have to give me the right to do so. I just realized.
They didn't put you as a host. Do we have tech with us that can put Andreas Kenma as a host?
Okay, Uri, maybe we skip for the next one, and then meanwhile I will ask him to do it.
Okay. Okay. So
Sorry,
Andreas,
no problem.
Andreas, are you already a host or not yet?
No, not yet.
Okay, so it will happen in the next 15 minutes, and in the meanwhile, Nimrod, maybe you wanna start with your presentation?
Yeah, sure.
Okay.
Can you hear me?
Yes.
Okay. Okay. So let's move this one. So hello everyone, I'm Nimrod Schwartz from the Hebrew University, and the title of my talk is Comparing the Drought Resilience of Different Tomato Rootstocks using Electrical Resistivity Tomography, ERT. Actually, this is the work of my student, Or Littig, and it's done with a collaboration of Muhaned, Mutez, Gauchau, and Andrea, and this is a great team, and I would like to thank them for the great work with them. And let's start. So the overall motivation for this work, I would say, is to develop tools for root phenotyping in order to develop eventually a drought resilience plant. So this is like the overall big motivation here. And there are many questions associated with root phenotyping. First, and this is of course an important question, is which root rates we would like to have in order to have a drought resilience plant, and it's not always clear which traits we need. And the second question, again, very challenging question, is how to measure those selected root features. And as I said, this is a great challenge because first, the link between root properties and drought resilience is complex, and there are many relevant root features like root length density, root architecture, root hydraulics, and so on and so forth. And the second challenge is how to measure these properties, and especially how to measure them in situ. Because first, the soil is opaque, so we cannot see. Second, it's extremely heterogeneous. The soil is heterogeneous, the root is heterogeneous, and also it's a very dynamic system, so it changes a lot in time. So what people typically do. So there are very nice laboratory tools for root phenotyping, and one of the main, I would say, laboratory phenotyping tools now is what is known as phenotyping platforms, like the one in the research center in Jülich that you can see here. So it's a very nice system of Rhizotrons, and you can do big experiments with many plants required for phenotyping. And you can image the root and measure architectures and other root properties through those systems. You can also use CT and MRI, but this is like more of a smaller scales. The main drawback of all of those laboratory and other laboratory methods is it's really hard to upscale those methods into what happened in the field, to field conditions. So the relation between what we see in the lab in this kind of nice box to what happened in the heterogeneous soil under various environmental conditions it's really complex. There are also field methods. Actually, two main field methods, I would say. The first one is chevelomics, it's called, and basically the idea is just to dig out the plant, wash the soil from the root, and start to measure the root architecture, and other root properties of interest. And the second one is using mini Rhizotrons. So this is plastic tubes that you install in the soil. You insert camera in these tubes, and you can image the root. Again, the main drawbacks is that you get local information about the root. Those methods are destructive, so and you cannot follow the dynamics. So in recent years, there is a lot of interest in using geoelectrical methods to do root research. I'm going to focus on one method, on electrical resistivity tomography. The main idea with the method is that you can image the subsurface electrical properties non-invasively. And using what we call geophysicists called petrophysical relationship, you can translate resistivity into water content. And this is, for example, what you can see here in this image, taken here in Rehovot in a wheat field that we monitor. And really, today, I think that we can say that we have success in applying ERT to investigate root water uptake, irrigation strategies, spatial pattern of root water uptake, and other processes related to root research and to agriculture. So the objective of the project I'm going to show you here was to compare the drought resilience of different tomato rootstock using conventional and geoelectrical methods. So let's see what we did. So what we did in the experiment is to graft M82 tomato plants on 3 different rootstock, and we did it in order to reduce the variability. So we have the same plant, M82, on different roots. Then we grow those grafted plants in a big plastic pots that you can see here. This is 70-liter pots. And we grow them using the best practice, so irrigation and fertilizers for 90 days. After this period, we stop the irrigation and we start with the measurements. So we measure quite a lot of variables. So we measure transpiration, the xylem water potential. We monitor the environmental conditions, so to calculate the VPD. We measure the water content locally using TDR, and using 128 electrodes. We also measure the electrical resistivity or the electrical resistivity tomography using the instrument that you can see here. So this is the DAS1 system. And eventually, at the end of the experiment, we also measured the root distribution, the shoot biomass, and other properties. So let's have a look at the results. So first, I would like you to look at this figure here. So what you can see here is the cumulative water loss from the pots for the tree plant that we had. So we had 3 different root systems, A, B, and C. And what is clear from this figure is that plant B transpire more water than plant A and C. So this is the first thing to observe. Here in panel A, you can see the flux from the leaf. And in panel C, you can see the flux to the root. So this is the transpiration rate divided by the leaf mass, and here by the root mass. And the important thing to see here is that plant B, the fluxes for plant B is higher than for the other plants. And this is interesting, I think, mostly because the amount of root for plant B is the lowest. So in this case, the plant with the lowest amount of root transpire more water, and the flux through this plant is higher. So let's have a look at the result from the water potential. So what you can see here is the xylem water potential as a function of time for the 3 species. So this is like the data, and we did some time series analysis. So to remove the diurnal cycles, and you can see here the trend. And what is clear is that plant A, so the water potential, this is the absolute value of water potential, so suction. So the suction of plant A is highest, so it is harder for this plant to extract water from the soil. So this is the first observation to make. And the second observation is that starting on around February 8th, we have a really sharp increase in water potential for plant B. And this suggests that the soil starts to be the limiting factor for the plant to take up, to transpire water. Okay, so we have the data from the transpiration, E, and we have the data from the water potential. So using this simple relationship, we can calculate the hydraulic resistance of the system. And you can see it here for the three species, A, B, and C. You can see the data, and you can see the fit to this simple equation. And here you can see the hydraulic resistance as a function of time for the three species. So two main things from this figure. The first one is that the hydraulic resistance is relatively constant for the at the beginning of the experiment, so as long as the soil wet. And actually, this really represents the resistance of the plant. So the soil is not limiting, and the highest resistors is the one from the plant. It might be the radial resistance of the root or the xylem resistance. I guess that this is the radial resistance of the root, and it's more or less constant and similar between the plants. And again, we can see that after more or less 10 days, we see this sharp increase in resistance for plant B. And this is actually the resistance of the soil. So really the soil started to be the limiting factor here.
Nimrod, what kind of soil was it?
It's a red sandy soil. So a soil from the coastal plains of Israel, real soil. Okay, so all of that was, let's say, data from the plant perspective, and let's have a look for the results that we have from the soil. So here you can see, and because of the time, I immediately show analyzed results. So what you can see here are map of the water content distribution in the three pots at two different times. And immediately you can see that, and this of course comes from the electrical resistivity. Yeah, so the electrical resistivity tomography, we measure or we map the electrical resistance, and using the TDR, the data from the TDR, we translate resistivity into water content, and this is what you can see here. So you can see that the soil dry out, and those red colors here. So the soil, of course, dries out. But trying to be a little bit more quantitative, what we did is to calculate the median resistivity and translate it into water content. So we have profiles of water content, and following Rao et al. Recent publication, we feed the data with a Gaussian function, allowing us to come up with two parameters. One of them is mu. This is the maximum depth of uptake. So this peak here in the function. And the second one is the distribution of uptake, which is the width of the Gaussian. So let's look at the results. So here you can see mu, so the maximum depth of uptake as a function of time for the three plants. And you can see that initially water are taken up from shallow layer of the soil. And with time, it's go deeper and deeper. And you can see that plant B take up water from the deepest layer. What else we can see is that the width of the uptake is more or less constant. So we don't have big changes in that. Comparing this to the root line density that you can see in this figure. So this is the percentage of root biomass with depth. You can see that most of the root, as expected, I would say, are located at the upper layer of the soil, and we have less root at the bottom of our big pot. And what is interesting, I think, is that first we can see that the root line density does not reflect the uptake distribution. And this is because we have the soil at the upper layer dries out, and root extract water from deeper layers. So this is really a clear evidence for compensation obtained with the ERT method. Another slide with figures similar to the one that you saw before. So we have the transpiration, we have the total root biomass, we have the depth of uptake and the root length density for another cycle of the same experiment. And what I want to say here is that we have similar results. So this is repeatable. This work is a work on progress, so we still have more experiments like that. And actually, even the phenomena of deeper uptake for plant to be here is more pronounced. Again, despite the fact that we have less root for that plant. So trying to summarize, first, I think that in this work, we are able to show that ERT is really a valuable tool for root research. Under drought condition, the depth of uptake is not correlated with the root length density because we have compensation. Drought resilience is not necessarily associated with root biomass. So, and this is something that we need to consider when we think about root phenotyping. So which root rate to choose. Exploring deep soil layers is an important root rate for drought resilience. That's it. I would like to thank for the funding agency and for you for listening, and I'll take any questions that you have.
Thank you, Nimrod. Just unmute yourself and ask a question.
So I can ask a question. Nimrod, you mentioned that you did some correlation between the TDR and the ERT to get those petrophysical functions. Mm-hmm. I wonder what about salinity, temperature, and other influencing properties about the water content and the ERT. Did you do something with them or any notes about it?
Yeah, so it's a good point. First, we did measure salinity, and it is not changing a lot during the experiment. But the key thing is that we did the correlation based on the data. So we measure every time that we took ERT measurement, we also measure the water content using TDR, and we use these data points to fit the petrophysical relationship, and not something else that we take to the lab and finding like the Archie parameters or whatever. So this is like really based on the data. I would say that this is data-driven fitting.
Thank you.
Thank you, Nimrod.
I have a very stupid question, if I may. So when I saw these pictures of those pots, you know, they had all those electrodes. I know you said over 180 per pot or something. Do they actually go into the soil, or how far do they go into the soil?
Yeah. So the penetration of the electrodes to the soil, it's some like 2 centimeters, some like that.
Oh, okay.
So it's 8 centimeter long electrodes, and they really penetrate to. The only thing that we want to obtain is good contact between the electrode and the soil. So in this method, an important step is to make sure that you have good contact resistance between the electrodes and the soil, and yeah.
Okay, cool. So you don't affect the soil too much then?
No, it's minimally invasive, and of course, as you can see here, this is and this is how typically it's applied in the field. So you really put your electrodes on the surface, and in this way. So this is why it's a non-invasive method. You don't really invade into the subsurface.
And with all these electrodes, the computational burden would be quite large, right, to actually make sense of the data?
Yeah, so of course there is a lot of computation behind it. So what you get from the measurement, just a table with numbers, and to obtain this type of map, you need to apply an inverse algorithm. And this is similar actually to what is happening when you In MRI, for example, everybody know that you for some reason need to image your body. So this is a similar algorithm, translate the data into a meaningful image.
Thanks.
Okay, thank you very much, Nimrod.
So Andreas,
please share the presentation. And yeah, Andreas.
Yeah, I'm there.
Yeah.
Can you see my presentation?
Not yet.
Okay.
Yeah.
Now it should be there?
Yeah.
Okay, perfect. Yeah, actually, so again, once again, good afternoon, everyone. Actually, it's great that Nimrod's talk now was first because he already introduced basically the principles of ERT, and I guess many of you already know this method because it's been increasingly used for imaging root water or soil water dynamics. And I'm going to talk about an extension of ERT, referred to as electrical impedance tomography, where also electrical polarization is measured and interpreted. And first, I would like to acknowledge my collaborator on this topic, Maximilian Weigand. So it's basically the two of us who, in different projects over the last years, worked on this in this field, yeah, in Bonn. Now, I can actually go quickly through the motivation because from what we just heard, we've seen that there is a demand for non-invasive imaging information on the soil root system. And one example is shown here, electrical resistivity tomography. So that's what just also a work that also Nimrod just cited, shows that we are able to delineate here zones of root water uptake through the effect of the changing water contact on the electrical resistivity. And one aspect with the electrical resistivity tomography method is that it shows less sensitivity on the root system itself. So if we address root zone questions, problems, then it's mainly indirectly through water contact. And so that's why the idea came up to, in addition to electrical conduction, so resistivity, to also look at electrical polarization. So physically meaning a capacitance of the root or the root systems, where to also recall the principle of the measurement method, where then we have a time varying current signal which is impressed into the soil, into the subsurface, and we measure a voltage response, where then the amplitude ratio of output and input basically refers to resistance or resistivity, and polarization gives rise to a phase shift between these two signals. So that's a measure of polarization or capacitive effect that is associated with the soil root system. And so the two main questions that we started with years ago with first projects, so my background is geophysics, so this has actually brought me into the field of biophysics, and which was new and exciting for me, by the way. And the two main questions were, so if there is electrical polarization of root systems or individual roots, so which information on root structure, and in particular also on root function, can we extract from polarization responses, from measurable polarization responses? And then the second one, of course, with a view to application of such a method at the field scale for root sensing, is can we utilize these signatures in a tomographic way, which is the prerequisite actually for an imaging method, and then to collect this information with spatial resolution in a non, or at least minimally invasive way. And to also recall the principle, I go quickly through this. We inject current at some frequency. Nimrod already mentioned that. So we have some current lines here for an example of a numerical example for a Rhizotron experiment, where some root growth is modeled in a very, very basic way. And that is actually the imaging result that we can expect from measuring a set of resistances, or now with conduction and polarization, a set of impedances, so including phase shift, that we can compute from a set of these transfer impedances. And you also see that we cannot expect, like with ERT, to spatially resolve individual root segments, but rather to delineate the spatial extension of the root system. And mathematically, we can describe the parameter that is imaged here as a frequency-dependent complex number, complex-valued electrical conductivity comprising magnitude and phase. And it's actually this phase that we're interested in particular, the frequency dependence of this phase. Yeah, and in early experiments, we investigated the imaging results of this method for different plants or root systems of different plants in simple Rhizotron experiments, where here, for example, an oilseed plant is placed in tap water, and we see that, well, with limitations in the spatial resolution, both conduction and polarization, here now measured as imaginary component of this complex conductivity, can delineate the root system. For the imaginary conductivity, so the polarization response, we also observe some significant frequency dependence. And this is actually what we are after in a way that that there is characterization potential in the spectroscopic response, which if we apply this ERT method in a spectral manner, which we combine with the spatial resolution benefits of a tomographic method. So it's basically merging tomography with spectroscopy. And if we look, for instance, now at for a pixel or a voxel of such an image at the spectral response that we recover, which is shown here, again, for an oilseed plant, which was placed in tap water and then just left alone over a couple of days. So this gave rise to nutrient deficiency over the days, and also an obvious degradation of the plant. And with this going on, we see then here the spectral response. So this is now versus frequency, the polarization versus frequency, that the spectral response changes over time, both in terms of shape and also in terms of strength. While for pixels from a zone where we have no roots, we do not get any measurable polarization response at all. And so this was providing evidence that we get something here with the polarization spectrum that contains physiological information, so that is sensitive to the physiology that is going on, whatever it is. But first of all, that holds promise to further looking into the method. And another example here at the lab scale in the Rhizotron is the monitoring. So it's again here the strength of polarization shown over several days in an Rhizotron experiment where we came up with artificial day-night cycles. So using a standard growth lamp, and you see here in light and dark, basically 12 hours with light on and with light off. And we were excited to see actually a cyclic behavior also in the polarization response. So another indication that obviously this polarization is sensitive to the physiological processes which are going on. This, of course, is temperature controlled, and and also it does not include soil at this point. Interestingly, we, by curiosity, we actually changed then here the artificial night to actually twice as long or even more, as you see here. And you see that there seems to, the root system, or at least the polarization response, seems to have some sort of memory effect, because it starts following the kind of general behavior, but then at some point again went up again the response, and then only with the lamp turned on again, we see this behavior, the decrease of polarization during the day. And of course, we also looked into, or many people, not many, but some people started looking into the mechanisms of these processes. And there's been lots of studies and literature on electrical polarization in porous systems, like in rock physics or soil physics, where some sort of polarization is related to electrical double layers at mineral pore water interfaces. Yeah, you see just such a scheme showing this double layer with some potential gradient, then which results in equilibrium between electrostatic and diffusive fluxes, basically. And similar double layers are also present at the root cell scale. And it's also more or less known that with, for instance, with ion fluxes, so thinking of ion uptake also of a root system as a whole, and then of course also at some cell scale, with ion fluxes across these cell membranes, these electrical characteristics of the double layer change. So in this case, actually with the uptake, the strength of the double layer would decrease, and that should have, in a similar way as for soils and rocks, an effect on the effective polarizability. So there's of course lots of room for research in that direction. Now, what's that?
Just continue.
Okay, I thought there was a question. Sorry. So my last slides show you an example of our first field experiments that we, of course, tried, because I said at the beginning that's the ultimate goal of the development of such a method. And we were lucky enough to have access to a Rhizotron field facility operated by the Research Center Jülich in Germany, where for this example, this is from 2018, we equipped the surface actually crossing through three plots with different treatments. Where maize was grown, we equipped this with electrodes at the surface. You also see here some electronic small units, which are all connected to some data acquisition system. I didn't have time to go into detail or talk about this, but it's actually also quite a challenge, the measurement of these signatures. And now showing you the result for this 2018 growing period, we decided to look at actually a spectral change of the polarization response. So I mentioned that we have this frequency dependence in the polarization, which can be measured by the imaginary component of conductivity. And we looked at the ratio conductivity at 1 kilohertz versus over conductivity at 1 hertz to eliminate some systematic influences, in particular associated with uncertainties in the error in the electrode positions. But this, from our investigations, seems to be a reasonable indicator for the presence of roots. And now this property, so after the imaging, after the inversion, this property is now plotted here in this cross section on top. And now start this animation. And on the bottom, you see basically the temporal evolution at different depths. And it's a bit patchy here across the 3 different plots. By the way, we have the one on the left, which was partly sheltered. So with an increased water deficiency or scarcity issue here for the root systems on that scale. Here in the middle plot, we have a double density of the maize. And I started again. You see that here it's May, June. We start seeing some signature, which, as I was saying, is an indicator for a presence of roots. So with this, of course, we can image within the spatial resolution limits the growth of the root system, and a bit quantitatively, it's better visible here in the bottom plot. where we see that it's actually first, these are, by the way, it's just artifacts in the animation. You see that we first get a response at shallow depth, as we expect, and then later this is followed by a response also at greater depth. And the nice thing and the important thing now for evaluating this method for its capability for the field application is that we had also rhizotubes installed where optical measurements were done and the images analyzed, and from those root length density estimates were inferred. And so we can here compare this independent information, the root length density vertical profiles plotted here for different times, for the 3 different plots, with the polarization response as a vertical profile, again in the same colors for the different days. And this is the first time that we could make this sort of comparison with independent data, being aware, of course, that also the root length dense estimates from these rhizotube optical images are subject to different influences. Nevertheless, it's promising that the qualitative behavior or the agreement is reasonable, showing basically this maximum rooting depth of the roots, and in particular with a greater rooting depth or larger depth extension for the plot which was under water stress. And with this, I would like to conclude. Take-home message is that we have, on top of the conduction properties of root systems, they also exhibit a spectral electrical polarization response with sensitivity to root system structure and function. And the function is actually the very promising aspect here, because methods are lacking that are sensitive to that. The mechanisms underlying the polarization effects are still poorly understood, in particular the spatial scales at which the polarization processes occur, and process-based models of polarization are missing, are still to be developed. So that is a very broad field and absolutely necessary to actually develop this into a quantitative method. However, lab and field experiments so far that we did show potential of the method for the structural and functional imaging, and if applied in a time-lapse manner, and that's the benefit of the method, also the monitoring of root systems. And I hope to see more applications also of this extension of ERT in the future. And with this, hopefully also the method, or in conjunction with other geophysical imaging methods, the method can contribute to an improved understanding of soil root processes. So with that, I would like to thank you for your attention, and also acknowledge contributions from other co-workers, in particular my colleagues Egon Zimmermann and Sander Heusmann at Jülich Research Center for the continuous support with EIT instrumentation. And some other names are mentioned here, and also projects funded by the German Research Society. Thanks a lot.
Andreas, I have a question. What do you think is the fraction of current that is flowing through the root system or the rhizosphere, maybe?
Well, this depends of the measurement design. So I was mentioning that we like to use as many electrodes as possible. And of course, one option is also to place directly electrodes at the stem of the plants. All the results that I've shown here, we did not do this. So electrodes were only in the soil. In the soil. So the current actually has to cross the quite resistive outer boundary of the root system to actually. So it's very limited. So I have right now no estimates in terms of current densities, but in terms of percentage, it's, or let's put it the other way around. The polarization comes from two contributions. So it's basically polarization, more of the rhizosphere. So basically the root system, soil, contact zone, and it comes from the interior of the root system. So that's more than a higher frequency cell scale polarization. And to see that, or how well we actually are able to see that depends on exactly how you say how much current we can drive through the plant. And in terms of percentage, it's only few percent.
Okay.
And of course, if we go through the stem of the plant, it makes it again more invasive. But there's a whole bunch of open issues, also methodological issues here, and also interpretational issues. So it's really must be seen as first results of the application of the method to the issue of root systems in an imaging way.
Question?
Yes, go ahead.
Hi, it's Lisa Wingate here. I'm going to ask a naive question. I'll just jump in. Is there a possibility with this type of approach to actually partition the contribution, also to look at mycorrhizal hyphae that are connected to root systems, or would the method somehow not work?
That's I cannot answer this at this point. So I try to stress that we have hardly any mechanistic, so process-based understanding how these signatures actually are built up. And of course, there is some analogies from electrochemistry and so on, but what really matters so the information that is contained in the spectral responses is certainly dependent or influenced by many factors. And I guess everything that has an effect on soil root contact will also influence the overall this overall macroscopic response that we measure. So to extract that and discriminate between different contributions, we certainly need calibration, like with ERT, where we need petrophysical relationships and so on. Nimrod mentioned the Archie equation. So we will need similar equations and independent laboratory studies to establish those also for the polarization. And then hopefully step by step we can discriminate or find ways to discriminate the different influences on this signature.
I can certainly see the next project, Andreas, with hyphae.
Yeah, well, yeah, I'm looking forward to that. So, but anyway, so nevertheless, I mean, there's many question marks, but I hope that it was that you also find it exciting that there is these signals that are measurable in a non-invasive way at the field scale and that are sensitive to physiological responses. So that is what we have shown so far. And this, I think, is a motivation also to now go further and address the different open issues.
Thank you, Andreas. We have to move on.
Professor Carminati, can you share your presentation?
Yes, I will do it. Do you see it?
Yes.
All right. So thank you for having me here. And so the scope of this presentation is to discuss how soil drying impacts stomata closure. And I will do it in a, let's say, theoretical approach using a model that I developed with Mathieu Javot, and with experiments run in combination with Nimrod Schwarz and Orlitic, and mainly run by Mohamed Abdallah, which is a PhD student in Beirut, where we used the same tomatoes that Nimrod described before. And here I will focus on two of them with contrasting root length. And the idea was to see if plants with different roots, they show also different stomata closure. So the motivation of this work is that soil drying is a primary constraint on transpiration globally. We heard it already at the beginning in the first presentation. but the mechanism by which soil drying impacts transpiration are still poorly understood, both in terms of soil physical question and physiological question. So specifically, my question here is to investigate how a decreasing soil water potential, psi-soil, impacts the leaf water potential, psi-leaf, the stomata conductance, and then the transpiration. And I will use a state-of-the-art hydraulic framework where I simplify the soil plant atmosphere continuum in 4 key hydraulic conductances. One is the hydraulic conductivity of the soil, so the water flow from the soil to the root surface. One is inside the root, from the root surface inside till the xylem. One is along the xylem and up to the stomata. Now, the important point is that all these 4 conductivities, they are variable. And they vary because of physical reason. This applies to soil and to xylem. And because plant adaptation, and this applies to the root anatomy, aquaporin, and adaptations in the root anatomy, and clearly in the stomata. Now, stomata are most probably the most important conductors in the system if you want to understand how much water is transpired by the leaf. However, it doesn't mean that the other conductivities are not important because they determine psi-leaf. And as psi-leaf is function of stomata conductance, then it comes clear that we need to understand how the conductivity of the soil, of the root, and of the xylem impacted the soil water potential, the leaf water potential, and then the stomata. So now, with this in mind, we try to develop a very, let's say, a model that is as simple as possible, which is a set of flow equation in the soil, in the roots, and in the xylem, where in each compartment, the flux is related to an hydraulic conductivity that is function of the water potential and a gradient in water potential. And the conductivity of both soil and the xylem, they are nonlinear and they decrease with the water potential. And here we took very, very simple example, just because we want to first describe this in a qualitative term. Now, if you solve this equation, you obtain such a surface. Now, in this surface, we are showing on one axis is the transpiration, the soil water potential, so decreasing from, let's say, saturation to decreasing to what soil scientists call the wilting point, around 1.5 megapascal. And on this axis, the leaf water potential. Now, this is a surface. It's, let's say, the physical space where the plant will move during soil drying. And the surface has an important, let's say, characteristic, which is that it has a linear part, which is here in green. And the linear part means that the conductivity of the whole soil plant continuum is constant. So there is no cavitation and the soil conductivity is not yet limiting. And the brown part where the conductivity drops. And this can be due to the xylem vulnerability, especially in this part when the soil is still wet. This was, let's say, a key point in the concept of sparring that was very similar to what we are describing here. And you have a part here where actually is the soil that is determining the nonlinearity of the surface. And we divided the linear and the nonlinear part by a red line here. And this line marks the hydraulic nonlinearity of the surface. And where the derivative of the transpiration as a function of leaf water potential, taking the soil water potential as constant, typically what happens during a day, when this slope starts to decrease. Now, the idea is that behind the red line, transpiration is, let's say, increasing transpiration is not convenient. So plants should rather close the stomata there, because if you increase it a little transpiration, it costs a lot in terms of leaf water potential. So the hypothesis is that the stomata regulation maintains plant in the linear zone of this surface and prevents plants from crossing the red line. After hearing the first presentation, I thought that it would be interesting to show the surface from the top view. where we're just looking at the leaf water potential on one side and the soil water potential on the other. And this is similar to the figure that Fulton showed, relating the pre-down leaf water potential, which could be psi soil here, and the midday leaf water potential. And stomata closure would bring the plants on this trajectory, which is similar to observation that that have been discussed in the first talk. So this concept has similarity to this observation. There is a consequence. There is a, let's say, consequent hypothesis, a corollary to the hypothesis that stomata regulation maintains plant in the linear zone, which is that stomata regulation then varies with varying soil hydraulic properties. It means that if this hypothesis is true, if I change the soil properties or if I change the root system, I will have a different stomata closure. And this is the hypothesis that we tested in the experiment that I'm going to show you. Is that the first hypothesis that, well, that stomata do close at the onset of soil hydraulic limitation. And the second is that stomata regulation, and more precisely the relation between transpiration and leaf water potential, should change with varying soil root hydraulic properties. And we tested this hypothesis using the same plant as Nimrod showed, using grafted tomatoes, so the same shoot, but with different root system, a short one and a long one. Now, what will be the consequence on the hydraulics? Well, the consequence on the hydraulics is seen in this picture. So the plant with the short root system, which is here is shown in the solid line, have a more marked non-linearity. Let's say at the same soil water potential, let's take this value. A plant with a short root system will deviate very quickly from the non-linearity, while a plant with a longer root system can sustain transpiration longer. Just simply because you average the transpirative fluxes over a larger surface area. So the fluxes of water in the soil are smaller, and so they're smaller the gradients in the soil. So longer root system should give a more linear behavior in this transpiration versus psi leaf space. And then I should have a later closure of stomata. So stomata for the short root system should close at a less negative water potential. Let's say here before 0.5 megapascal, while with a longer root system should close later. Now, Moanad did experiments measuring the transpiration rate and the leaf water potential using a root pressure chamber, which pressurize the root system so that we can maintain the leaf turgid and so the stomata open. And this is the result of 3 plants at different water contents. The water contents decrease from blue to red. And what you're seeing is that as the soil dries, these curves, they become more and more linear. And you have some difference between the short and the long root system. You see that the long root system tends to maintain these results a bit more linear. So you can sustain a higher transpiration at a more negative leaf water potential. So this was what was expected. Now, we also measure the transpiration reduction during soil drying with non-pressurized plants. And the results are shown are the red squares shown here. And in this figure, we show many things. It's a bit complicated. So we show the results before. Then we have the solid lines. These are the fitted. So this is the, let's say, these are the simulation that we run to fit the, let's say, the open circle. And so fitting the hydraulic space, we expect this red line, this stomata closure, the red line indicated here. And you see that the measurements match pretty well with the model in both the short and the long root system. So this figure shows 2 things. It shows that, first of all, it's true that stomata close at the onset of hydraulic nonlinearity, basically when the black lines start bending. And the second thing that it shows, and now I will show the same figure, but simply comparing the transpiration response between the two root systems. And so the second result is that changing the root system, that here changing basically the gradients in soil water potential around the roots changes also the relation between transpiration and leaf water potential. So the result of this experiment is that different root systems trigger a different stomata versus leaf water potential relationship. It means also that stomata adapt to varying soil moisture condition in a flexible way, and there is no predefined stomata versus leaf water potential function. So actually, in summary, the loss of soil hydraulic conductivity around the roots is the trigger of the closure of stomata. And the consequence of it is that stomata regulation is not simply a function of leaf water potential, but it depends on soil and root hydraulic properties in a, let's say, in a predictable way. So with this, I would like to thank, well, first of all, Mohamed Abdallah for running the experiment. Here is the whole team developing and using the root pressure chamber, which actually was invented by John Pasura. And this is a collaboration with Israel through the joint German-Israeli water technology research program with Nimrod. Thank you a lot.
Thank you, Andrea.
Again, a soil scientist that will say that everything is the soil.
Well, I'm a little biased, I admit.
I have a question that kind of combines, I think, all the speakers we've seen so far. So both Tony and yourself, Andrea, suggested that there is a difference in stomatal regulation in respect to the soil volume. And I think that Andreas and Nimrod showed us that the soil volume, or not the soil volume, sorry, the root volume is likely to change in response to the water deficit itself. Mm-hmm. So the assumption that it's, let's take a small root volume or a large root volume doesn't exist, right? While it dries, probably to explore other volumes of the roots. It expands its root, other volume of the soil, expands its roots to more available water. So how will that kind of change the model?
Yes, I mean, this is, I think it's a very good point, Uri, that you bring up, because this concept that I showed, it's, let's say it's for a relatively short drying period. And we're mainly looking at, let's say, a short time regulation of fluxes, which is through the stomata. So this day, let's say they operate at a high temporal resolution scale, but soil dries slowly. And if you go in the field, it takes time. And during this time, you have an additional plasticity, which is the one of how the roots are growing. And now probably how the root grow in relation to how much the shoot grows. And this may explain also, well, may need it to link the results that we showed now and the one that we saw before with Nimrod. So yeah, I think there is an additional temporal scale and an additional coordination between above and below ground that I'm not, I didn't talk about, and actually I don't know it.
Okay.
Actually, Andrea, I would have a question, a curiosity mostly on this experiment. The long and the short roots are the same genotype, so one was more advanced, or you make it grow more, or there are two different genotypes that are characterized by the different root size?
No, there are two different genotypes. They are the same age. Yeah.
Thank you.
Uri, we need to move on. Thank you, Andrea.
Thank you all.
Thank you. We have Celia now. Celia, are you ready?
Yes.
Your screen?
Okay, can you see?
Yeah.
Okay. So thank you very much for inviting me here. In this, well, you can see the now. Okay, well, continue with the topic of Andrea's talk. I will talk a bit about recent results in olive seedlings, where we showed that the disconnection between the root and the soil affected the canopy conductance. Also, I will show new images from microcity, microcity images on roots or how these roots behave during soil drought. And also, I want to discuss a bit how to link these new insights with the precision agriculture and how we can optimize irrigation, or how we can optimize water use in agriculture. So I don't think I have to spend much much time in this, because I think everybody knows that. But well, stomata are very important, are crucial for the plant response to drought. They control, they regulate the water use and productivity. So the gas exchange, between through stomata, which is known as GS, it is to know it is very important under agriculture perspective, and most importantly, what is triggering the stomatal closure, because it will give us the idea of the loss in productivity. Graphically, if we represent this, it will be like this graph, where we can see that we have three phases. In the first phase, stomata open and close, mostly depending on environmental and light conditions. Then, as the drought increase, stomata close, and to reduce transpiration, to reduce this water loss, and also to avoid damage in the xylem. When this continues, cavitation appears in the xylem, and this goes to more extreme events, and if we cross the point of no return, this is fatal for the plant, and the plant likely dies. So in agriculture, although it is important to know the cavitation resistant thresholds, we mainly move between these two phases, between the one where stomata are mainly functioning and the one where stomata close. No? So if we look at this response, this declining in GS, we may think that the hydraulic conductance is somehow, or the GS is somehow independent on what is happening in the hydraulic conductance. So according to the hydraulic approach, and this is also very well explained by Andrea, we can predict the limits in transpiration, in plant transpiration, knowing the vulnerability to cavitation in the xylem of the plant, and also knowing the vulnerability of cavitation in the soil. So that means that any alteration of this continuum may affect to GS and limit plant transpiration. So we did an experiment in olive seedlings, where we partitioned the different components of the hydraulic pathway between the soil and the leaf. We used a combination of rehydration techniques to measure that, and rehydrating the plants from the outside and monitoring the changes in stem water potential, we were able to measure the root hydraulic conductance plus the soil root interface. And then rehydrating internally the plants, we were able to measure the rest of the components of this hydraulic pathway, which are K root and the K shoot that include leaf and stems. If we show the results, so these are the result of this experiment over a water potential level of down to minus 4.5 megapascal. We saw that there were not changes neither in the shoot nor in the root hydraulic conductance, and only in the root hydraulic conductance plus this soil root interface, we found a decrease in the level between -1 and -2.5 megapascal. And this had a drastic impact on the whole plant hydraulic conductance. And also it was so we saw this result also in the limitation at the canopy conductance. So we thought that something was happening in this soil root interface, and we explained it according to many others results, that this could be due to root shrinkage, so damage in the root hairs, and root and changes in anatomical in the anatomy of the root. So 2 months ago, we conducted an experiment at the synchrotron in Paris, and to see how these roots behave during soil drying. In this case, we used my seedlings that were grown in 3D pot, in 3D printed pots with these syringe barrels, where so this facilitate the microcity scans where the root grew. And now with these high quality images, we can add them to our graphical representation of plant response to drought. And we can see how at high water potential, the root was perfectly in contact with the soil and the root hairs were fully turgid. As soon as the soil dehydrated, the root hairs started to shrink, and then the root start to shrink and to form these cortical lacunae. And finally, if the drought continues, all these events increase. But we still need to conclude this experiment, because we need to quantify the shrinkage and this disconnection, the lacunae formation, and also to add the characterization of the stomatal response of these plants and the measurement, the quantitative measurement of K root. But I think this adds more evidence about that we need to consider also the interaction between the roots and the soil to fully understand how plant responds to drought. But this doesn't mean that this event occurs or triggers stomatal closure. In fact, I think it's more like what Andrea was saying, that is more like a temporal thing, because we know that at diurnal basis, angiosperms have evolved during millions of years to respond very quickly to changes in atmospheric drought by producing very rapidly ABA and closure stomata. So in the end, everything ends at the stomata, because they integrate everything. They connect the soil with the atmosphere. They control or they regulate the water use and the productivity. So as I think in agriculture, we need to have this target, which is GS, to optimize the water use in our orchards. And in fact, this is what we are actually doing in Seville, in Spain. We are extensively characterizing the physiology of 6 fruit tree species. And also we are using different sensors to automatically monitor GS, which are subflow and leaf to pressure sensors. And we demonstrated already that this is possible, the automatic monitoring of GS using this sensor in olive, where we monitor diurnal GS using subflow sensors and maximum GS using leaf to pressure sensor. So we have now excellent techniques to measure this water stress in real time. And as concluding remarks of my talk, understanding stomatal function and how they close is fundamental to comprehend how plants respond to drought. Any alteration of the hydraulic conductance of the continuum between the soil and the canopy can limit, so it works more like a limiting factor in a seasonal time frame, but we know that in a diurnal time frame or more minutes time frame, ABA is closing stomata in the leaf. And finally, we need to link all these insights to optimize irrigation, to optimize water use in agriculture, starting by monitoring GS to optimize productivity and saving water. So I want to thank all the funders that make this possible and all the people that collaborated in this project, and also to you for listening. Thank you.
Thank you very much, Celia. Anybody want to ask a question? Unmute yourself and ask. So I'll ask a question.
So Celia, you mentioned the resistors and the potentials and the conductivities. What about capacitors? Do you think that we should have some capacitors in this interesting model?
Yes, of course. Capacitance is very important in this continuum, I think. And what I think is that this time events is more due to these capacitors, and these capacitors works like enabling the plant to resist more or less the droughts, and to use the water differently. So yeah, definitely we need to add also this capacitance within these processes.
Celia, by any chance, I'm very curious about your Solel experiment. Did you try to rehydrate the plants and see if the roots go back to their original shape? Or...
Yes, we did it, but well, I didn't want to spend. I mean, it is still under construction, all this work, because it was like just 2 months ago, and well, we didn't have time to analyze all the data. But yeah, we rehydrated the roots, and they very quickly reconnect to the soil. So, and depending on the the water potential that the roots were, they take longer or shorter time to fill again this gap. So if you can see the gap that is created between the root and the soil, it reconnects again with the same volume, with that. And now the question is, okay, but how this is related with the recovery of the plant? And because if you rehydrate a root very damaged, it can recover its volume, but probably will not transport water anymore, because there are too many gaps between the xylem and the soil surface that it will not transport water. So you will need to, I mean, the plant will need to grow new tissue to do that. But yes, we rehydrate also these roots.
Celia, how difficult it is to conduct those experiments in Paris? I mean, you need to pay them. You need to. What is needed there?
That's a difficult question, because I was not leading that experiment, but yeah, they funded your project if the project is good, but I think it's very competitive. But I mean, I don't know. I just went because well, I could make this collaboration with Sylvain, with Hervé Cochard, with José Manuel Torres, with many people there. So, but I don't know how this works.
INRAE has a platform there, and so through collaborations with people on INRAE, you can get beam time, and you can also get some funding for people to attend. So
yeah.
Okay, Celia, thank you very much.
Thank you.
And we have Craig Broderson with us. So, Craig, do you want to share your screen?
Yeah, can you make me big? Does that work? Can you make that big? And I'm in front of it. Is that okay? Instead of sharing the screen? Or I can do it the other way too.
No, we will not see it in the platform. You need to share the screen, please.
Okay. Yeah, I'll move back. No problem. All right. So.
Great.
That's okay there? Okay, perfect. All right, well, thank you. I want to first start out by thanking Uri and the rest of the program coordinators for inviting me to talk. I've been looking through the list of participants and seeing some of the talks over the past couple of days, and it's been really great. I also wanted to thank the couple of collaborators that are involved in this project, Andrew McElroone at UC Davis, Martin Buda, who's the research scientist now at the Czech Academy of Sciences, and Carl Vint, who helped us do the NMR imaging. And so the goal of this project was really a simple, to answer a simple question that's been bothering us for a really long time, and it sort of arose out of a conversation that we had at a conference in Germany a number of years ago, where Carl Vint was there. And the question, and I know that after looking at the participant list, I know that there's many of us that have used this equation, the Hagen-Poiseuille equation, to estimate the potential of flow through a circular conduit that we use to approximate what's going on in the xylem. And the idea here is that we've been measuring hydraulic conductivity through stem by attaching them to a hydraulic apparatus and then pushing water them. And then we can also figure out how good of a job that this equation does at approximating the flow rate through a stem, right? Through these essentially circular pipes. And so the idea here is that the main thing that we typically consider is the diameter of the conduit. And so based on looking at this equation, and this has been used in a number of different iterations, but we'll just use this one because it's probably the most common, is that there's three important parts of the equation. One is the diameter of the conduit, the viscosity of water, and then of course the pressure gradient. And so the diameter of the conduit ends up being very important because a small increase in the diameter leads to a very large increase in the flow rate. This is on the right side of the screen there is an image from taken from Mel Tyree, the Tyree and Zimmerman book, just showing that a 40 micron diameter conduit has the same hydraulic conductance as 16 small ones or 256 even smaller ones. So a large increase in flow just from a small increase in the diameter of the conduit. And we can use this equation to go in and look at something like, there's two different examples here. These are two common hardwood species that we find in our forests here. There's a sugar maple on the left, an Acer species, and then a Quercus on the right. You can immediately see that there's a difference. The one's a ring porous species, the oak on the right, and then on the left is a diffuse porous species. And you can see that there's a large difference. These are taken at roughly the same magnification, taken from the Inside Wood database from North Carolina State University and Elizabeth Wheeler's work. And you can see that immediately, just at the same magnification, that there's a really large difference in the diameter of these conduits. And so we might want to then go in and measure, using the Hagen-Poiseuille equation, go in and measure the diameter of each of those different conduits, and then predict what the overall conductivity is, the potential conductivity is for that, for those two different stems. And so here is on the right side of the screen is just an estimate, right? So we've got maple with a conduit diameter typically less than 100 microns, typically somewhere in 40 and 50 microns is where most of the vessels end up. And then you have oaks that often have really large diameter vessels, 250 microns at maybe the most, maybe 300. And you can see at the individual conduit level that there's certainly a hydraulic advantage of having these really large diameter conduits. I did a rough estimate on those two previous images, and you can see the predicted flow rates down in the lower right-hand corner, right? So that the maple should be at a disadvantage compared to the oak if we just take this, the diameters alone and use the same pressure gradient pressure drop across each of those different vessels. And so while the viscosity of water is probably important in real life, right, in the field, due to thermal changes that take place throughout the day, certainly the one thing that we don't often take into account in using this equation to predict the hydraulic conductivity through a stem or through a cross section of a stem is the pressure gradient. And so pressure gradient is probably, if all of us being plant scientists here, would recognize that there is very likely going to be non-homogeneous pressure gradients on the inside of the stem, simply because of where the upstream and the downstream components of the pathway and what they're experiencing. And so a very simple illustration is that if you have sun on one side of the tree, maybe the south side of the tree in the northern hemisphere is going to be getting more sunlight than the north side of the tree in the northern hemisphere, then there's going to be potentially more going on here, simply because the plants are open, the leaves are opening their stomata, and the water potential gradient is therefore higher. And so this has been done with sap flow sensors, and you can see that there are differences around the tree, and they sort of respond, the tree as a whole responds as you might predict. Right. And so in reality, there's uniformity across, if you look at the pressure gradients inside the stem, even though when we do these predictions of how much water you can push through a stick, those are always done in a way where there's a uniform pressure gradient. And the question is, how far away from reality is it when we're using this equation? And so we know that there's going to be internal differences in the pressure gradient inside the stem. So we use this Hagen-Poiseuille theory to predict flow based on conduit diameter alone and just use a uniform pressure gradient. And so the question is, what does that look like inside the plant? And so to answer that what seemingly a very simple question is we need two things. So we need measurements of in-planta flow for individual vessels, right? To say that do the large diameter vessels in a stem actually transport more water than the narrow diameter stems based on theory. And so we need to do those measurements, we used this NMR velocity flow imaging that was done by Carl Wendt and Jülich. And then we were able to take that same stem and scan it with the high resolution micro CT imaging, similar to what Celia just described in France, but we do ours at the Lawrence Berkeley National Laboratory in California. And that allows us to put together a network map of all the vessels and all the different connections, because there's growing evidence now that the internal network connectivity is extremely important in the network dynamics and how water is actually flowing through a stem. And so part one is the in vivo flow part. And so what we have here in the red image, sort of in the center, is the water content map. And this is basically just telling us in the cross section of a grapevine stem that's approximately, I don't know, I think is about 8 millimeters in diameter, or 5 millimeters in diameter. So it's a relatively small plant. You can see an example in the lower right-hand corner of the plant in the very specialized magnet NMR system that they have there in Jülich that Carl helped us with. And so he took a Cabernet Sauvignon plant, put it inside of the magnet, and then was able to provide this NMR velocity flow map, which is on the right there. And the blue areas are showing regions or vessels where there is low flow, and then as the flow increases, changes from green to yellow to red, where we see high rates of flow. So the problem with the NMR velocity flow imaging is that the resolution sort of limits our interpretation, because the pixel sizes are 300 to 400 microns. The largest diameter vessels that we're seeing grapevines of this size are somewhere on the order of 200 microns, 150 to 200 microns. So you've got one large pixel that's representing potentially many vessels. And so the idea then is that we can merge this type of data set where we have the actual flow of water going through individual vessels or groups of vessels in a transpiring plant, and then use micro CT imaging, which has a much higher magnification, a higher resolution image, in order to basically align the two and overlay them together. All right, so this is something that we've been working on in pulling out the vessel network parameters for quite a while. So we published this paper back in 2011 or 2010, where we went in with a number of different methods, so including light microscopy and scanning electron microscopy, to basically figure out, we've got, in this case, our micro CT image on the right here in this panel B, and then panel A, the scanning electron microscopy image of the exact same spot in the stem. So we can determine what the threshold distance is for determining when two vessels are connected. And using this method, we're able to go in and identify every single vessel that's in the stem, give it a unique identification number, and we can determine all sorts of different things that are really difficult to measure otherwise, including the total pit area between stems, how many vessels are actually connected within the network, within the three-dimensional volume of tissue. So we end up with something that looks like this, where you're sort of you're staring down the middle of the grapevine stem, in this case, sitting inside the pith, and each one of those blue lines represents a vessel, and the vessel diameter is represented by the diameter of those pipes, and then every single one of those yellow dots inside this image is the position of a connection or a node where there's a connection between two adjacent vessels. And now that we have this information, we can use it to predict flow, and we can use the Hagen-Poiseuille equation, for example, and apply a pressure gradient across this network to see how well how well our model, the digitally extracted xylem model, predicts the actual flow that we're seeing in the images, the NMR velocity flow images that Carl gave us. And so a lot of this work was done by Dr. Martin Buda, who was a postdoc in my lab at the time. He's now at the Czech Academy of Sciences as a research scientist there. And we basically, Carl took the stem from the NMR velocity flow imaging, put it in a tube, sent it to us in Berkeley, and then we scanned the exact same area. And then this sequence of images here shows our attempts at aligning the two images and overlaying them. And so the first one, again, is our water content map. The second one in B, in the middle panel there, is showing the actual flow rate of the water through the individual vessels. And then panel C in the upper right-hand corner is showing the micro CT image, and showing in yellow in particular, this yellowish orange region, showing the region that we decided that we needed to use because the vessels that were in that outer ring of vessels are not fully developed and we're not actually transporting much water at all, showing that they were still under development as the plant was continuing to grow. The bottom right image and the bottom series of images there is our attempt to line up the two, and allows us to get the high resolution image and figure out what the vessel diameters are and all the network connections that are present in that the NMR image above, right? So we get these two overlap together, and then we try to basically use the 3D Xylem network model in order to predict and find good agreement with the in vivo flow model. So what we did is we basically push water through this through the 3D Xylem network model and try to predict the flow rate. And so what you're seeing here is the measured flow rate up on the upper left-hand corner in panel A. And so this is what Carl measured in the NMR velocity flow system. We're seeing the large diameter vessels, which are typically clustered at the top and the bottom of the stem. So we're just looking at the cross section of the stem here, and we've masked out all the vessels that aren't really contributing to flow, presumably because they're not fully developed. And what we see is where the large diameter vessels are on the top and the bottom, which we call the axial, the dorsal and ventral parts of the stem, and these lateral parts on the side are the parts that feed out to the developing fruit and tendrils, for example. And so that's our measured flow rate. And if we use the uniform pressure scenario, as we typically do in the Hagen-Poiseuille equation, what we find is the modeled flow in the middle sort of looks okay. But if you take the image on the right-hand side, which shows the model residuals and basically showing us where we're over predicting or under predicting the flow rate based on the uniform pressure gradient, we aren't able to get a very good fit, suggesting that just using a single uniform pressure gradient, every vessel in the cross-sectional area of our model getting the same pressure drop from the top to the bottom does not allow a good fit. And so when we do an independent pressure scenario where we allow the software to basically go in, and the modeling software to go in and pick the pressure gradient that is going to be needed in order to get good agreement between the real flow and the modeled flow, that only then do we actually get a really good fit. And so our model residuals go close to zero, meaning that there's very good agreement between the two. So this leads to some potential issues in our use. One of the things a couple of things that we found is that on the left-hand side here, we've got the pressure differences between the vessels from the mean, and showing that there are large differences in the water potential of the xylem sap moving through different parts of the network. So we get there are axial gradients, sort of which we would expect going from the top and the bottom, but there are also transverse pressure gradients of significant size. So, there could be a pressure gradient over just a very short distance, a few hundred microns of perhaps a megapascal or more, which is sort of hard to think about, but it sort of it intuitively makes sense given on what we know is going on above downstream in the plant. So one of the outcomes of this is on the right-hand side here, where we have our vessel flow on the y-axis in micrograms per second, and then we have our vessel radius on the x-axis. And the blue line that's running through there is our Hagen-Poiseuille equation and our predictions. And so for small diameter vessels, it appears that the Hagen-Poiseuille equation actually does a pretty good job of predicting what the actual flow rate is in this particular plant. But most of the large diameter vessels are actually underperforming compared to what you would expect. And so it appears as though the knowledge of the Xylem network topology, where all the connections are, how they're distributed, and the different series of resistances that are imposed by having this complex network end up being really important sort of design elements. So design in air quotes there. And what's also interesting is that vessel connections redirect about 15% of the flow through the narrow diameter vessels, which is sort of not what you would expect if you just went in and looked at a single cross section of the stem, ignoring all of the intervessel connectivity, and also ignoring the physiological activities such as stomatal opening or closure that's going on upstream or downstream or upstream in the root system. And so that's sort of where we're at with this, and it sort of we think it raises a number of different questions regarding how we actually use these measurements. And if you have a stem that you put into a hydraulics apparatus to generate a vulnerability curve, for example, and you apply a uniform pressure gradient across that stem, what is that? Is that actually telling you what's really happening? So and kind of gets into these ideas of the differences between conductivity and conductance of the stem. So how much the stem could potentially transport versus how much these vessels actually are. So with that, I think I'll just leave it there for questions.
Thank you, Craig. Anybody want to ask a question?
Yep, may I?
Of course.
Sure.
Great presentation. Thank you for sharing. I was just wondering, so with your imagery and your estimation of the flow resistance, so it's a coupled question. Can you also estimate the resistance that is contributed by the pits at the vessel ends? And sort of connected to this, did you actually test that your estimation of the resistance in each vessel is correct? Because you could just take a segment, right, and apply a homogeneous pressure gradient across it, just as we do when we measure hydraulic conductivity, and then use the micro CT to actually, the MRI to measure the flow velocities then, and this would then give you sort of, it would eliminate this uncertainty about the actual pressure gradient, and would allow you to test whether your estimation of the flow resistance distribution among the vessels is correct, right?
Yeah, that's a great question. So to the first question, the idea is that the network model is based on a number of different input parameters, and so it will get the 3D Xylem network extraction process and all that automated or semi-automated software that we have developed it will identify the vessel diameters, which is obviously an important component. Most of the stem segments that we're working with don't have vessel endings. We think that a lot of those are located in the nodes rather than in the internodes, and so there's some there are some caveats there, and that we're looking at a very small piece of the entire network. And in this case, Carl was able to image a very the same spot that we were looking at, and so we get a prediction about what's going on in that position. And then the other parts that go into the flow estimations are either the resistivity of the pits that has been measured in other systems, or measurements that we've made from just pushing water through stem segments. Let's see, I'm trying to think. The other part is something that we are working on. The current restrictions for travel and things like that have limited the ability of our to follow up and do those experiments where we take a stem segment and put it into the system. With the NMR, it's always a little bit tricky because nothing magnetic. All non-magnetic materials can't be inside the magnet, so it makes it a little bit tricky to do some of these experiments. But it's definitely something that we're going to follow up with.
Thanks.
Yep.
I have a question, if I may. I love the talk, Craig. Thank you very much. Consequences of these findings. Is it possible that somehow then this underperformance of the larger conduits somehow contribute to set a limit to maximum vessel size?
So, yeah, that's an interesting question. So I think that there's, the vessels that we weren't. So there were really large diameter vessels, in particular around the outer part, right? The most recently developed vessels. And so I think that there's probably some expectation that those vessels are certainly going to eventually become fully functional and transporting water. But a lot of, I think what's going to be determining the vessel diameter is both as the plant certainly gets older, they tend to produce larger and larger vessels up to a point. there are the amount of water that's available and the other resources that are available to the plant during development are also going to influence the maximum vessel size. But it seems, at least in this, so we need to put all of this within a very small conceptual box, and that we're only talking about a couple of plants in the study at a very specific point in developmental time. So I think this is, we now have the tools to sort of go in and answer exactly that question. In that we've got the NMR velocity flow imaging, and that type of system works thanks to some of the work that Carl's been doing. And then now we have the modeling side complete. And so it's sort of, we're in a really nice position now to sort of go in and answer questions like that.
Okay, thank you, Craig. Thank you very much. So we have the last speaker for today, Jonathan. Are you ready? Do you want to share your screen?
I assume that Jonathan is your student.
It appears to be so.
Jonathan, we cannot hear you. You need to unmute.
You can hear me?
Yes.
Great. Okay. So today I want to show you my results in my first project in my PhD. I did it in this vineyard in Israel last summer. So embolism resistance is considered as a static trait. Here you can see an example. Fontes measured the dynamic water potential. Here in blue is pre-dawn, in red is midday, and compared it to a static value of a turbo loss point and P50. And from this result, he assumed that the plant, this tree from the Amazonian forest had cavitation. Because the minimum water potential here cross the threshold of the cavitation. This approach is not unique to this paper, and when you upscale this approach, here is a chart. He took a lot of species and compare the minimum water potential that he took from the field as a dynamic measurement. He compared it to the P50 that he took from a database, a static value. But there is a few evidence for a seasonality in direct traits. Here you can see a paper from Charrier. He found in 8 cultivar of grapevine that the vulnerability curve of the stem decreased from July to September in 7 bars. The turbo loss point that is frequently correlated to the vulnerability curve to embolism resistance is also had this phenomena. He over the season, the turbo loss point in many plants decrease over time. Finally, Martorell took 2 grapevine cultivar with 2 water treatment and compare the P80 to the turbo loss point over the season, and he found a nice correlation. So thank to Celia. I don't need to
Explain.
Thank you. Explain you the basic model of how plants react to water stress, and it's probably true for rapid water stress. But we hypothesize that over the season, there will be a shift in the water potential values to a lower values. So the two loss point will shift with the stomatal conductance and the vulnerability to cavitation to a lower water potential values. This strategy allow to the plant to sustain a more drier environment towards the end of the season. To do that, we collaborate with our friend, Dr. Isha Netzer from Ariel University. He had experimental vineyard with irrigation treatment for several years. So today I want to show you just two treatments, the super irrigated with hourly irrigation every day, and the no irrigated treatment without any precipitation all the season. My results. So here you can see the no irrigation treatment. The midday stem water potential decrease over the season. And in May, we measured the vulnerability curve in the leaf. And instead to assume that it's static, we measured it twice more. And we found that embolism resistance in the leaf is dynamic over the season. To check our fully hypothesis, we took branches from the no irrigation treatment 3 times in the season, and we dehydrated them while we measured the stomatal conductance here in the solid line and vulnerability curve in the dashed line. And also we had the 2 loss point of those leaves. We did it in May, in July, and August. As we expect, there is we found a shift in those 3 traits along with the season. So we found a correlation between hydraulic traits that maintained for all treatments and throughout the season. As you can see here, the blue is May, green is from July, and red is from August, and the shape represent the treatment. I want to mention that in many papers you see this correlation between the embolism resistance to total loss point, but we did it in one species and over time. Also, until now, in those data, our data and the Charrier, we don't really know for sure if the change, the decrease in the vulnerability curve throughout the season is because of environmental factor, the drought stress, or development. So therefore, we had the second treatment, the super irrigated treatment. This is the midday stem water potential. And also we had the methodological data from this vineyard. And as you can see, the temperature, the VPD, and the evapotranspiration was the highest in the highest value in May. This is very typical to Israel. We had here in the beginning of the season a couple of heat waves. So because of that, when we found, here you can see the blue box plot, that the super irrigation also decrease is a vulnerability to cavitation in the leaf over the season. And when we assume that it's because of the development and not because of any environmental stress. So, but in grapevine, the leaf size decreased along with the branch growth over during the season. So in all of those dates, we measured the youngest fully mature leaf. So in May, the leaf was the bigger than the other dates that get smaller. So we want to check if maybe if this phenomena related to the leaf size that can be represent the xylem architecture. So what we did, here you can see the vulnerability curve of the leaf from the no irrigation treatment. We took it in May and August, you already saw that. And what we did, we took another leaf, the May position, we took it in August, and we found that the leaf, the embolism resistance of those leaves shifted to a lower water potential that's more likely to the leaf in August. These results suggest that all the plant shift to a lower water potential, and not just the leaves that emerge later in the season. So now after we saw that a fully mature leaf can change its embolism resistance, we want to maybe to show how. So we suspect that maybe pit membrane thickness is related to, because there are several papers that correlate the pit membrane thickness to embolism resistance. So to do that, we collaborate with, we took 3 times along with, or 3 times in the season, branches and send them to Germany, to Dr. Steven Jansen, and he measured them for us. This is our plants. This is how it looks like. And we found that also pit membrane thickness is also dynamically and change along with the season. And when we correlate the P12 to pit membrane thickness, we get a nice correlation. But as you can see here in the blue, this is the leaf from May, and they are a thin outlier. So what is the difference in those leaves? In those leaves, the electron density of the pit membrane was much less than the other dates. This is why we think that after the pit membrane thickness can explain the shift in the embolism resistance just after the electron density that we don't know exactly what is it, is high. So, and also, as you can see here, the same leaf, we took it in May, and in August, the pit membrane thickness shifted. So my take-home message, the P50 can decrease over time, and it is not a static value. And there is a coordinated seasonal shift across multiple hydraulic traits, and pit membrane thickness or density also was dynamic throughout the season. This is our article, we published it last month. Thank you. It was my first lecture in, so thank you. Thank you. This is all the people who work with me. Smadar and Yossi in the field. This is Ishai. Ilana and Steven do the anatomy measurement, and this is Uri, my supervisor. Woohoo! Thank you. Question?
Thank you, Jonathan.
Thank you. Kol hakavod.
I hope that every lecture will end with a paper from a student.
Anybody has a question for Jonathan?
I'll ask a question. So any thoughts on those May leaves and the, what does the low electron density of those pits mean? I mean, just give me a wild guess.
I think it's not fully. It's full of water. It's just the beginning of the season, and the leaf just emerged. It's 1 month ish, and it's not sticky yet.
So you're saying it's immature, but not in a sort of leaf developmental way, but in some sense a seasonal way. There's some kind of seasonal maturation of the plant.
Yeah,
It's not just distance from the meristem. That's interesting.
I think this the components that need to flow throughout this leaf. This is why it's changed over time, because the flow, it's all the time flow and component stick there or something. Maybe calcium.
Okay.
Last question, maybe?
Jonathan, just a curiosity. The pit membranes that were analyzed are from the leaf or petiole or stems?
From the petioles, the between the petiole to the midrib.
Okay, the attachment of the leaf petiole.
This is what usually measured. Stephen is an expert.
Okay, thanks.
Okay, thank you very much, all of you, for a great session. It was really fun for me, even during those days with all the corona around us. It was fun to see you all, to hear you all, to get questions. I encourage all of you to participate in the final session. There are sessions that are going on now, but the final session will be in 7:30. We will have there several great scientists, Ratan Lal and the lead scientist of the UNCCD. So I think it's a very important to participate in the final session. Thank you all.